Good morning all. I hope you understand my all previous videos. Now it's time to move on our next chapter from the Maharashtra State Board Textbook Standard 9, chapter number 8, Useful and Harmful Microbes. What the name suggests, useful and harmful microbes means in lower classes you are studied about the microorganism, either in the 8th class also you studied about the different microbes, that virus, bacteria, fungi is there, algae, protozoa. Okay, so all these are the microorganism and these all are responsible for causing certain diseases in the human body or in the animal or on the plants, or sometimes few microorganisms are useful to us also. So in somewhat they are useful and they are harmful. So in this chapter we have to study about their uses and their harms. Again we are going to study about their morphological characteristics, how they are looking, where they are present, what are their uses, what is their nature. Okay, and we are again studied about antibiotics. The uses is that bacteria or that microorganism also useful for making the antibiotics, means medicine. So let's start with our chapter, that is the useful and harmful microbes. The question is asked here, can you recall? That question is based on your previous knowledge. What is mean by microbes and what are their characteristics? So what is microbes? Microbes are a tiny germs or very small microorganism that we cannot see by our naked eye, but we can easily see them by under a microscope. So it is a microbes. We cannot see them by our eyes, either it is present in air, water or on land also. Everywhere there are presence of the microbes, but still we are not seeing them due to their very tiny size. Their characteristics, it is their characteristics that they are too small, invisible, they are bearing some uses and some harms also. Okay, so all these are the characteristics of their microorganisms. Again, next question is that how did you observe the microbes? How did you observe the microbes? We can observe the microbes or microorganism under a microscope. Now what is a microscope? Microscope is the instrument that used to enlarge the image of the object. माइक्रोस्कोप ये एक ऐसा इंस्ट्रूमेंट है जो कोई भी ऑब्जेक्ट की इमेज को एनलार्ज करता है. उनमें लेंसेज होते हैं, अलग अलग लेंसेज, जो कोई भी टाइनी ऑब्जेक्ट का इमेज और बड़ा करते हैं. उसको फोकस करके हम उसको और बड़ा देख सकते हैं. इसलिए हम ये जो माइक्रोब्स है जो हमें इजीली आंखों से नहीं देख सकते, हम उन्हें माइक्रोस्कोप के अंडर देखते हैं. You are familiar with different type of microbes which are all around us but cannot see them with our eyes. In what way are they related to our everyday life? Yes, we are familiar with different types of microbes that present in nature and are near about places. Okay, but we cannot see them by our eyes. They are invisible. So how they are related with our day to day life, कि वो हमारे डेली लाइफ में कैसे फिर इंटरफियर करते हैं, या वो हमें हमारे साथ वो क्या रिलेशन में है, कहाँ प्रेजेंट रहते हैं, उनकी हमें हेल्प होती है या वो सिर्फ हार्म करने के लिए होते हैं. तो चलो देखते हैं ऐसे ही कुछ माइक्रोब्स. फर्स्ट वी आर गोइंग टू स्टार्ट और फर्स्ट टॉपिक, दैट इज़ अ यूजफुल माइक्रो जो हमें फ़ायदे के माइक्रो ऑर्गेनिजम्स है वो. इन दैट वी स्टार्ट्स विथ फर्स्ट बैक्टीरिया, दैट इज़ द लैक्टोबैसिला. The name is new for you, but as I am going to tell you about the whole history of that lactobacilli, it is a very familiar to you and it involves in your day-to-day life also. See how that lactobacilli present in the buttermilk or curd. You know what is the buttermilk? Tak. Curd means yogurt, dahi. So that lactobacilli is very familiar to us, but we don't know the name of that particular bacteria, or even we are not seen to them. So like that, different microorganisms are also involved or also related in our day-to-day life. So first we are seeing, that is the lactobacilli. Now here again one question is there, if you have to observe the microorganism, सो व्हाट यू डू फर्स्ट, अगर आपको लैक्टोबैसिलाई भी देखना है तो आप सबसे पहले क्या करोगे? ओके, जस्ट सी. Okay, now first thing is that you have to make a smear of a drop of fresh buttermilk on a glass slide. Your first thing is that if the two sample is provided to you here, buttermilk also and curd also, what you do here, just take either buttermilk or you can use curd. Just take a drop of buttermilk from this container, put drop on over a slide. It is a rectangular glass slide. Okay, normally it is available in the lab for the practical purpose, so it is a slide. Just take a drop of buttermilk on a slide, uniformly spread it on a slide. Just spread that drop on a slide so we can make a smear of that drop. It is known as smear. What we have made here, that is a smear. We just take a drop from that test tube and uniformly we spread that drop on a slide, so it is a smear. That word is given in your test book, so understand this. What is smear? When a drop of any sample, we spread uniformly on a slide in a particular area, it made a smear. So now your smear is ready. Now what you have to do, just let the spear to dry. Spear को आप dry होने देंगे. What you do after that, just stain that smear with methylene blue. अब क्या करोगे कि ये जो स्मीयर आपने फॉर्म किया है इसे आप वॉश करोगे. किससे? स्टैन करोगे मिथिलीन ब्लू. ओके, यू जस्ट स्टेन इट विथ methylene blue. Now one question is arise in your mind कि what is methylene blue? It is one of the stain means color. ये एक ऐसा कलर है जैसे हम लोग डाइज से कपड़े हेयर कलर डाइज आते हैं, वैसे ही ये एक इट इज़ वन ऑफ द टाइप ऑफ डाइज जिसे हम साइंटिफिक लैंग्वेज में स्टेन कहते हैं. ये स्टेन ऐसे रहते हैं जो माइक्रो ऑर्गेनिजम्स को कलरफुल बनाते हैं. सम माइक्रो ऑर्गेनिजम्स आर प्रेजेंट इन नेचर, दे आर कलरलेस, बट you cannot see the colorless thing. That's why if we have to observe such thing or such microbes, then in that case we have to stain that particular slide so we can easily see that bacteria. So after drying of the smear we stain it with methylene blue. तो हम लोग क्या करेंगे, स्मीयर ड्राई होने के बाद उसको स्टेनिंग करेंगे. स्टेनिंग मीन्स कलर करेंगे. किससे? मिथिलीन ब्लू. इट इज़ लिक्विड इन स्टेट. ये आपको ऑलरेडी मार्केट में अवेलेबल मिलता है, कोई भी केमिकल शॉप में मिलेगा मिथिलीन ब्लू जो लिक्विड रहता है ऑलरेडी. सो अब इसको पूरा कवर करोगे मिथिलीन ब्लू से. आफ्टर दैट पुट कवर स्लिप ओवर इट. अगर आप फिर से लेबोरेटरी में देखते हो, इंस्ट्रूमेंट्स में अगर उसके चेक करते हो, तो आपको एक ऐसी ही प्लास्टिक की बनी हुई स्मॉल स्क्वायर शेप की कवर स्लीप मिलती है जो बहुत ही थीन रहती है. एंड दैट कवर स्लीप वी पुट ओवर द स्मीयर दैट बियर्स अ स्टेन. ओके, जो आपने स्टेन किया है उसको आपको फिर से वॉश भी करना है. ठीक है, वो कंप्लीट स्टेन के ऊपर कवर स्लिप नहीं रखनी है. मिथिलीन ब्लू से स्टेन कर दिया आपने, स्टेनिंग के बाद आपको उसको वॉश कर दोगे और वॉशिंग के बाद आप उसके ऊपर हाइड करोगे क्या, कवर स्लिप. ठीक है, कवर स्लिप ने से आपने अपना स्मीयर जो है वो हाइड कर दिया है या पूरा कवर कर दिया है. आफ्टर दैट द नेक्स्ट स्टेप इज़ दैट जस्ट पुट दैट कंप्लीट स्लाइड अंडर अ माइक्रोस्कोप. उसके बाद आप क्या करोगे, ये जो स्लाइड आपकी प्रिपेयर हो गई है आफ्टर स्टेनिंग, आफ्टर पुटिंग अ कवर स्लिप, ये जो स्लाइड है आप कहाँ रखोगे, माइक्रोस्कोप में. इन दैट just see in your textbook that smear under, observe that smear under 10x objective of the compound microscope and then into the 60x objective. So what is that 10x and what is that 60x objective? So to enlarge the object or to enlarge the object of this whatever we are taken on the slide, we just focus that slide under a microscope. But the three types of the objective are given in that microscope: 10x, 60x and 100x. Okay, so these are the different objective that already provided in the microscope, but on our level, or just to see the microorganism, we just use the 10x and 60x objective. So first we focus the microscope in a 10x objective. After that we rotate that part of the microscope and set on the 60x objective, so we can easily see the particular bacteria that present on a slide. Next, now you focus the slide also. What is your observation? When you focus the slide under a microscope, you notice there the blue rod shape, because the color of that methylene blue is blue. So whatever the bacteria or the lactobacilli are present in that buttermilk, they pick up that stain. ये जो stain का color है वो pick कर लेते हैं, and that's why they seen as a rod shaped bacteria, but these are having a blue color. Okay, so they are exactly the lactobacilli, and they are a bacteria, and they are very minute, rectangular in shape. Okay, sometimes they are rectangular in shape also, and that lactobacilli are anaerobic bacteria. Anaerobic means they can survive in absence of oxygen also. You note the meaning of that word in your textbook. Anaerobic means in the absence of oxygen, and they can produce energy also without the use of oxygen. ये ऐसे बैक्टीरियाज रहते हैं जो अनएरोबिक होते हुए भी वो एनर्जी प्रोड्यूस्ड करते हैं. मीन्स इन द एब्सेंस ऑफ ऑक्सीजन ऑल्सो दे कैन produced energy. Now next question that is given in your textbook, can you recall what is the question? How the yogurt made from milk and what exactly happened in this process? So next question is कि how yogurt made from milk, की जो curd है वो milk से कैसे बनता है, उसमें exactly क्या होता है. Just take one of the container here that bearing a milk. एक container हमने लिया है जिसमें हमने milk लिया है. So now if we have to convert that milk into yogurt, what is the procedure for that? Now here the lactobacilli bacteria that already present in milk. Okay, the lactobacilli, it is a type of bacteria that already present in the milk. Now what is the use of that lactobacilli in the milk in converting the yogurt? So and you know the sugar is present in the milk, that is the lactose. So the lactose sugar is present in milk. So what the lactobacilli exact do here? That lactobacilli convert that lactose into lactic acid. Okay, what is the function of lactobacilli? That lactobacilli converts the lactose from the milk into lactic acid. So now it is a type of acid, okay, that released in the milk. In milk, the lactose converts into lactic acid. Now the acid is formed here in milk. The next is that process that converting the lactose into lactic acid, it is called the fermentation process. As a result, what happens here? Due to the production of lactic acid, the pH of the milk decreases. Now what is pH? As you study, what is pH exactly? Okay, you know the pH scale. The pH scale ranges from 0 to 14. Okay, it is a pH scale. Just reminder this thing. Okay, it is beneficial for the higher classes also. So it is pH scale that you helps in science 1 also. pH scale that starts from 0 to 14, and middle number is a 7. So from 0 to 7 means 1 2 3 4 5 6, and here 8 9 10 11 12 13 and 14. So the number ranges from means 0 to below 7. It is a acid, and the number ranges from 7 to 14 means from 7.1 to 14 they are a base. Okay, and the substance that belonging to pH 7 only, it is the middle number. Okay, below 7 all are the acids, above 7 all are the bases, and the substance bearing a pH 7, they are neutral in nature. Okay, so the pH ranges from 0 to 7. All substance are we called acid. The substance having a pH 7.1 to 14 all called as a base, and the substance having the pH 7, they are only neutral substance. So here what happened after formation of the lactic acid in the milk, the pH of the milk decreases. Okay, now the pH going to decrease, pH कम होने लगा है means जो pH ओरिजिनली pH जो था मिल्क का वो कम होने लगा है, मतलब धीरे धीरे यहाँ किसकी क्वांटिटी बढ़ रही है, यस एसिड की. सो ड्यू टू द फॉर्मेशन ऑफ द एसिड इन द मिल्क, हियर द कोएगुलेशन ऑफ द मिल्क प्रोटीन ऑकर्स. कोएगुलेशन ऑफ मिल्क प्रोटीन मीन्स मिल्क प्रोटीन्स क्या होते हैं, कोएगुलेशन मीन्स क्लम्पिंग. व्हाट इज द मीनिंग ऑफ कोएगुलेशन, क्लम्पिंग. क्लम्पिंग मीन्स उसके गड़े बनने लगते हैं, वो एक दूसरे को जॉइंट होने लगते हैं. इट कम्स टुगेदर फॉर्म अ क्लम्प्स. तो इनमें मिल्क में क्लम्प्स फॉर्म होने लगेंगे. ये क्लम्प्स एक दूसरे को अटैच्ड होके रिमेनिंग वाटर इसमें रहता है. ओके, कुछ क्लम्प्स फॉर्म हो जाएंगे और यहाँ कुछ वाटर फॉर्म होगा. दैट वाटर is called that present after clumping, it is called a whey. Okay, the water is called a whey. So the milk protein are separated from the constituent of the milk. Now the milk proteins are coagulate and it separate from the remain constituents that present in the milk like water also. So due to that, the milk that originally present in that container now changes to curd. Okay, it is the complete procedure of making the yogurt from milk. And after making the yogurt, that yogurt having a specific test, that is a sour test, and that sour test is due to the formation of lactic acid. And due to the low pH, due to the decreasement in the pH, or due to the acidic condition, whatever the microbes or the harmful microbes that present in the milk become destroyed. The harmful microbes become destroy or kill. Next, use your brain power. In that, some question is asked to you and you have to find the answer of this question. All are very easy and just based on your daily routine. So why do doctor advise you to have yogurt or buttermilk if you have indigestion or abdominal discomfort? Now I explain all the thing here, ki what happens in the milk, how the yogurt is made from the milk. Okay, so just think on that topic and give or find out the answer of this question. कि sometimes you also experience that thing that doctor advise you to take the buttermilk or yogurt if you have indigestion. Indigestion means अगर आपको proper digestion नहीं हो रहा है, abdominal discomfort या फिर stomach एक वगैरह का problem है, पेट दर्द का problem है, तो आपको क्यों डॉक्टर एडवाइस करते हैं? Just try to find the answer from this topic. If you don't understand, ask me. Sometimes yogurt becomes bitter and froth up. Why does this happens? Sometimes yogurt become bitter and froth up. You know, when the curd is, or the yogurt means curd, so when the yogurt is stored for more days, it gives a more bitter taste. So why does this happens? क्यों वो ज़्यादा कड़वा होने लगता है, क्यों उसके ऊपर फ्रोथ अप मीन्स फेस आने लगता है, क्यों ऐसे कन्वर्जन उसमें होते हैं? Third question, which different milk products are obtained at home by fermentation of cream from milk? Which are the fermented products we can made at home from the cream of milk? तो मिल्क की जो क्रीम रहती है, या तो फिर फर्मेंटेशन टेक्निक से हम लोग घर में कौन कौन से डिफरेंट प्रोडक्ट्स बना सकते हैं. नेक्स्ट, डू यू नो extra information given here to improve your knowledge. What is mean by probiotic yogurt and other food stuff that are popular nowadays? Nowadays what happen, if you are going to the bakery, then you observe there a probiotic yogurt. अब हमने नॉर्मल योगर्ट तो देखा था, नॉर्मल कर्ड तो हमने देखा है, इवन अलग अलग प्रोडक्ट का ब्रांड का भी देखा है, लाइक हल्दीराम, अमूल. बट फिर ये प्रोबायोटिक योगर्ट क्या है? क्या ये कोई ब्रांड है? नो, इट इज़ नॉट अ ब्रांड. ये प्रोबायोटिक योगर्ट एक हेल्दी प्रोडक्ट है, योगर्ट है, हेल्दी योगर्ट है. फिर अब इसमें क्या प्रेजेंट रहेंगे? ओके, और ये हेल्थ के लिए क्यों अच्छे हैं? Useful microbes from like Lactobacilli are added to these eatables. Such eatables are healthy because they kill the harmful bacteria like Clostridium in the alimentary canal and helps to improve our immunity. तो ये प्रोबायोटिक योगर्ट्स क्या है और ये अब ज़्यादा पॉपुलर क्यों हो रहा है? क्योंकि whenever we are making the yogurt, biotic means living microorganisms. So whenever we are making the yogurt at industrial level, okay, for the distributional purpose, तो उसमें हम क्या करते हैं, जब ये योगर्ट बनाते समय हम उसमें एक्स्ट्रा लाइव बैक्टीरिया ऐड करते हैं. अब कौन से बैक्टीरिया ऐड करोगे आप? लैक्टोबैसिली. ओके, तो ऐसे यूज़फुल माइक्रोब्स आप लैक्टोबैसिली जैसे माइक्रोब्स वो ईटेबल्स में ऐड करते हो. तो वो ईटेबल्स क्या होते हैं, जैसे हमने पहले ही पढ़ा है कि वो लैक्टोबैसिली से क्या होता है कि जो हार्मफुल बैक्टीरियाज है जो मिल्क में प्रेजेंट रहते हैं वो ऑलरेडी किल हो जाते हैं. तो इसलिए हम जब ये प्रोबायोटिक योगर्ट्स बनाते हैं तो हम ऑलरेडी लैक्टोबैसिली की क्वांटिटी एक्स्ट्रा क्वांटिटी में हम उसको ऐड कर देते हैं वो ईटेबल्स में. एंड ड्यू टू दैट एक्स्ट्रा क्वांटिटी ऑफ द पर्टिकुलर बैक्टीरिया और पर्टिकुलर माइक्रोब्स, दैट ईटेबल्स बिकम्स मोर हेल्दी एंड दैट ऑल्सो हेल्पफुल फॉर किलिंग ऑफ द हार्मफुल बैक्टीरिया. तो ये हेल्दी भी होते हैं और हार्मफुल बैक्टीरियाज को किल करने के लिए हमारी हेल्प भी करते हैं. अब आपने अगर ये आंसर सर्च किए तो आपको एक और मिलेगा, जैसे कि क्लोस्ट्रीडियम. दैट क्लोस्ट्रीडियम इज़ द हार्मफुल बैक्टीरिया दैट प्रेजेंट ऑन आवर एलिमेंट्री कैनाल, and due to the lactobacilli that clostridium also kill and that helps to improve our immunity. Next are the uses of lactobacilli. The first use, the various milk products like yogurt, buttermilk, ghee, cheese, shrikhand, etc. can be obtained by the fermentation of milk. As the same process we carried out to make yogurt from the milk, like that by using the same fermentation technique or by adding some other microbes also, or by using the other techniques also, we can make the buttermilk, ghee, cheese, shrikhand also with the help of milk. The second use is that lactobacilli fermentation is useful for large scale production of cider, cocoa, pickles of the vegetables. Again, that lactobacilli fermentation also used in making a different product like cider. It is the alcoholic drink made from the apple juice. Just note the meaning: alcoholic drink made from apple juice. Cocoa, that cocoa either in available in market in the form of powder also, that you know the cocoa powder is very familiar to you, that you made chocolate cakes from that cocoa powder, or that cocoa powder also used in making the milkshakes. Okay, and that cocoa powder made from the cocoa seeds, pickles of vegetables also. The third point, lactobacilli and some other useful microbes taken together are used to treat the abdominal discomforts. Yes, the lactobacilli and other microbes also we can use to treat the abdominal discomfort. अगर स्टमक एक वगैरह है तो उसको क्योर करने के लिए हम लोग लैक्टोबैसिली जैसे कुछ माइक्रोब्स को यूज करते हैं. Fourth use, leaven fodder offered to domestic cattle like cows, buffaloes, and is fodder fermented with the help of lactobacilli. Leaven fodder means चारा, जो हम कैटल्स को चारा खिलाते हैं, लाइक काउज एंड बफेलो को जो हम खाने की चीज़ें देते हैं. ओके, दैट leaven fodder and that leaven meaning it is given in the form of dough. जैसे गूंदा हुआ आटा जो फॉर्म में रहता है, बट वो आटे को हम लोग फर्मेंट करते हैं किससे? विद द हेल्प ऑफ लैक्टोबैसिला, बिकॉज इट इज़ वेरी इसेंशियल फॉर द डोमेस्टिक कैटल्स टू यील्डिंग देयर और टू इम्प्रूविंग देयर मिल्क यील्डिंग कैपेसिटी। दूध देने की जो कैपेसिटी रहती है उनकी, वो इंक्रीज़ करने के लिए या इम्प्रूव करने के लिए हम लोग उनको ये लीवन फॉडर ऑफर करते हैं। द लैक्टोबैसिलाई फर्मेंटेशन प्रोसेसेस इज़ यूज टू मेक द वाइन एंड सम टाइप्स ऑफ ब्रेड। तो ये लैक्टोबैसिलाई हम लोग फिर से वाइन्स बनाने के लिए, ब्रेड, केक्स, ओके, तो ऐसे अलग अलग डिफरेंट बेकरी प्रोडक्ट्स भी बनाने के लिए हम लोग लैक्टोबैसिलाई को यूज करते हैं। Now just try to search the answers. How many different industries depend upon the lactobacilli bacteria? Yes, you know, as we studied all that thing, you can easily find out the answer. Which type of the cottage industries and factories can be started in the areas with abundant milk production? मतलब जिस एरिया में बहुत ज़्यादा मिल्क प्रोडक्शन है उसमें कौन से कॉटेज इंडस्ट्री या फैक्ट्रीज स्टार्ट हुए हैं? Just find out the answer of these questions in our next video. We are going to study about the rhizo